Hi, we're excited today to be talking about the Global Pandemic Prevention and Biodefense Center. We've got three incredible panelists today that are incredibly accomplished, and they're going to share part of their story with us and their interest in this center and why it's important as we look at preventing pandemics in the future. Let me briefly introduce them. First of all, we've got Dr. James Crowe. He's the director of the Vanderbilt Vaccine Center. He's also the Ann Scott Carroll Professor of Pediatrics, Pathology, Microbiology, and Immunology. He is a board-certified pediatric infectious diseases physician, and he's a viral immunology researcher. His laboratory studies of the human immune response to infection for a wide variety of major human pathogens includes many emerging infections as well. He's an elected member of the National Academy of Medicine, and he's been the recipient of numerous investigator awards. A number of the vaccine candidates and human monoclonal antibodies developed in the course of his research have also been tested in phase 1, 2, and 3 clinical trials in humans. Dr. Crowe, we're pleased to have you today. We also are very pleased to have Dr. Nahid Badalia, and she is the founding director of the Boston University Center for Emerging Infectious Diseases and Policy and Research, and is an associate director of the National Emerging Infectious Diseases Laboratories, affectionately known as the NEEDLE, which is a state-of-the-art maximum containment research facility at Boston University. She is also a board-certified infectious diseases physician and an internationally recognized leader in highly communicable and emerging infectious diseases with clinical, field, academic, and policy experience in pandemic prevention and response. Over the last decade, Dr. Badalia has designed and served as a medical director of the Special Pathogens Unit, a medical unit designed to care for patients with highly communicable diseases in a state-designated Ebola treatment center. She has prior and ongoing experience in health system response to pathogens such as H1N1, Zika, Lassa fever, Marburg virus, and COVID-19 at the state, national, and global levels, including medical countermeasure evaluation and diagnostic positioning, infection control policy development, and healthcare worker training. Dr. Badalia serves on state, national, and interagency groups focused on biodefense, priority setting, and development of clinical care guidelines and medical countermeasures research. She served as a subject matter expert to the U.S. Center for Disease Control and Prevention, the Department of Defense, and the World Bank. She's published in Nature, Science, New England Journal of Medicine, and other prestigious journals, as well as in the press, including the Atlantic and the Time magazines. Her work has been featured in documentaries by National Geographic as well as Nova, and you see her on NBC and MSNBC as a medical contributor. Welcome, Dr. Badalia. And third, we have the Honorable Andy Weber. He's an independent consultant and a senior fellow at the Council on Strategic Risks. He's dedicated his entire professional life to countering nuclear, chemical, and biological threats and to strengthening our global health security systems. Mr. Weber's 30 years of US government service included 5.5 years as President Obama's Assistant Secretary of Defense for nuclear, chemical, and biological defense programs. And he was the driving force behind the Nunn-Lugar cooperative threat reduction efforts to remove weapons-grade uranium from Kazakhstan and Georgia and nuclear-capable MiG-29 aircraft from Moldova to reduce the biological weapons threats and to destroy the Libyan and Syrian chemical weapons stockpiles. In addition, he coordinated US leadership at the international Ebola response for the Department of State. Mr. Weber is currently a strategic advisor to Ginkgo Bioworks, and he's a consultant for DARPA, Lawrence Livermore National Labs, and others. Honorable Andy Weber, thank you for joining us here today. And these are the three panelists that we're going to discuss this Global Pandemic Prevention and Biodefense Center with today. As quick background, about 1.5 years ago, as COVID broke, a task force formed in Greater Washington. That task force was made up of 50 leaders across industry, academia, government, and community. And in that task force, they evaluated critical responses to make sure that as we emerge ultimately from COVID, that we're in a better position than we were before. One of the ideas presented was presented by Dr. James Crowe, And it actually called for getting in front of preventing pandemics. This captured the attention of those 50 leaders. They listened to Dr. Crowe, and they then unanimously adopted the pursuit of the idea that he presented as a critical global response for preventing pandemics. So Dr. Crowe, let's start with you. Tell us a little bit about how you came up with this notion of a Head 100 and this ability to get in front of pandemics. What led us to that thinking?
Yeah, thanks for having us and addressing this topic. I think it's a topic that's on everyone's mind. We don't want to go through a pandemic again or a major epidemic, and what can we do about it? So my own experience has been working in the area of monoclonal antibodies, and we have worked for years to respond to epidemics that occurred. So bird flu, like H5N1, or chikungunya outbreak, and Zika outbreak, and Ebola outbreak. And each time these outbreaks occurred, we mobilized our team to make human monoclonal antibodies that could be used to prevent or treat those infections. But these processes take months to years to go from dead stop until you have a drug in hand. And each time we did this exercise, we realized we were really too late to make an impact on the epidemic that occurred. And in fact, these things are so regular, they're about every 18 to 24 months. By the time we were finishing one, the next one was already happening. And it just became sort of frustrating. I was thinking, wow, what could be faster than what we're doing now? And the only way to get ahead of these things was to do it ahead of time. That's where the ahead idea came from. And we just started saying, well, what if we made antibody prevention or treatment drugs for all of the microorganisms that were likely to cause epidemics, already causing epidemics or likely to cause large ones. And you can make a large list of those, and that's what we did, and we embarked on trying to make them. But as a single lab, we can't really do that. We can do the discovery piece of it, but I realized in talking with many potential partners, this was going to have to be a major effort that involved a public-private consortium. We need government, we need philanthropists, we need foundations, we need academics, we need commercial parties who are manufacturing. And there really was not a place at the time to do that kind of work. So I've just been looking for a partner to sort of bring this vision fully forward into practice, and that's where we are now.
Thanks, Jim. And so this cross-sector approach is intriguing. Dr. Bedelia, tell us a little bit about the center that you run and why that cross-sector collaboration is so important as we contemplate pandemic prevention.
You know, Jim nailed it. I think that a vision like that, a prepared world, requires that connectivity. So I actually work for two organizations, as you know. One is the BSL-4 lab that we mentioned, which, you know, for the last decade working in that environment, you can sort of see the importance of maximum containment laboratories for a portion of that type of work. Limited model lab space that's in maximum containment that can, for certain pathogens, provide the type of information that can take the research to the next level, to be able to give us more accurate, you know, live pathogen information, both immunologically, but also for animal models and things like that. But one of the things that we faced at the lab is, you know, we are always at this precipice of being involved in public health emergencies. I'm you know, at my baseline as Jim is, I'm a healthcare worker, you know, and most of my involvement in pandemics, first and foremost, has been as a frontline healthcare worker. And that sort of connection of when crisis happens, how do you connect the research to the frontline healthcare worker, to the patients? And the center that I currently direct, the BU Center for Emerging Infectious Diseases Policy and Research, comes from this understanding that if an organization like Needle is the wet lab, right, of the work around diagnostics, vaccines, and therapeutics, then a center like SEED is the dry lab. It is that connectivity. It is the connective tissue that deals with public health research, you know, ethics, modeling. You know, what we've realized is the importance of science communication. And so our center is organized around 4 important priorities. One is governance, a lot of things that I think Andy is also involved in, which is really looking at national, local, and international governance and policies and how they help and hurt sometimes, right? Regulations and other things. resilience, looking at community level resilience, community level pandemic preparedness, and that includes capacity building, as well as healthcare level resilience. What can we do to create that ability to take on a surge or to deal with the depth of care that's required for the highly communicable pathogen? Innovation, everything from social innovation to technological innovations and diagnostics, and lastly, trust, which we found is so important for uptake of new technologies. Even basic, public health measures, non-pharmaceutical public health measures. So governance, resilience, innovation, and trust is what we're really focused on. And the importance of a center like the one that we're talking about is that, there's three parts of getting it right. One is being fast, one is being flexible and connected, and the third is sort of having that level of trust and connectivity and transparency. And the network that the center is building is going to be well positioned to do that.
Super, thanks. And Andy, as Dr. Bedelia is talking about that trust and that integration, again, collectively, you as advisors, and Jim, you as the chief scientist for this AHEAD 100, have basically laid out an open and collaborative approach. Andy, you've spent a lot of your time serving in the government. How does this cross-sector collaboration really work? Does it drive the benefits that Dr. Bedelia is talking about with speed and flexibility? Why is this the right approach for us with the AHEAD 100?
Yeah, I agree with what Jim and Naheed said, that this integration function is really essential across sectors. There are so many examples of this. The most recent really stunning success is Operation Warp Speed, where you had a full partnership between the Departments of Defense, Health and Human Services, industry, academia, working together to act quickly and make this incredible number of vaccines quickly available to the American public. So that's the right model, is working across sectors as a consortium to move faster, because in epidemics, pandemics, time, months, days, weeks, it can be an eternity. It makes the difference between our ability to nip an epidemic in the bud or have a situation like we're in now, where it's spread around the world and it's really too late to isolate it.
And what all of you have kind of accomplished with your respective organizations during COVID is incredibly remarkable. And, I think when this journey started, there were a lot of questions about, can you actually prevent a pandemic? And now I think these last months have really proven that you can get on the other side, right? You can get on the pre-side of boom, if you will, for pandemic prevention. And so antibodies are kind of central to this center. Jim, there's the common understanding of antibodies, I think we've seen through COVID-19, are pretty limited. And so why are antibodies the focus of this center? Why is this the technology and capability that we need to focus on?
Yeah, when you think about the things you can do for infectious diseases, there's non-pharmaceutical things, masks, PPE, and so on, distancing. And we've deployed those to, better or worse effect in the current pandemic, but we're working through how do you do that. And then you have antiviral drugs, like small molecule drugs. They're generally not particularly good for acute infections, because by the time the person shows up, their virus is already peaking, and having an antiviral effect is very challenging, especially when people are already in the hospital. And then you can have vaccines that completely protect. They're very important, but they often take a while to develop. And one of the critical limitations about vaccines is you can't use them effectively in most immunocompromised people, as an example. So there are about 3 million such persons in the United States who really cannot respond adequately to vaccination, and they're also at an increased risk. And then you have people at other comorbidities, heart disease, autoimmunity, all sorts of other organs involved, and those people don't respond as well to vaccines. So there's this population in which you could do prevention or post-exposure prophylaxis with antibodies. And antibodies are an interesting hybrid, in my opinion, because now that we have long-acting antibodies, it looks like they essentially can serve as vaccines. Recent trial that we just did with COVID, it's predicted by the pharmacokinetics that you would get protection for as much as a year. So that's a vaccine-like effect when you get a single shot and you get protection for a year. The antibodies also can be used after you've been exposed. That's called post-exposure prophylaxis. So if we were all in a room together and I were infected and just inoculated all of you, if you knew that within the first few days, you could get antibodies, and that would interrupt the event. And then finally, many of the antibodies that have been deployed in the current outbreak are being used essentially for early treatment, so post-exposure or early treatment, and they work there too. Whether or not they'll work for therapy, I'm not sure, because they face the same barriers as any antiviral of acute infection. It's probably very difficult when infection's late. So you have this wide spectrum of antibodies that can fill in the gap where small molecules and vaccines just can't do that mission. And I think the innovation around long half-life turns them into almost vaccines too. So very exciting technology, but as you said, in the current pandemic, we sort of beta tested using antibodies, and we learned a lot, but we didn't make a major effect so far because they had to be infused at an infusion center by IV, and that just turned out to be a limiting resource. It's very difficult to get people into the infusion centers. Most centers can only do about 10 people a day, and that just is not the scale we need. So we're going to have to be able to inject these things with a needle. I think that's one lesson we learned. And the long half-life is a new opportunity that we hadn't really thought about. How would you use that? How, when, and where? When you've got a vaccine and a long-acting antibody, where would you deploy these things? So I think the current outbreak is a learning episode where antibodies did not, or at least to date, have not had a major effect. But using these lessons, we could think about the next time how to do it better.
And so, Dr. Bedelia, you work a lot with the training, the policy, the enablement. And as Dr. Crowe just talked about, infusion going basically to intramuscular and other forms of administration. How big of a jolt does that put into the healthcare system as we think about a response? Is that complicated? Is that easy to kind of assimilate? How do we think about that?
I'm going to layer on something that Jim just mentioned, which is that timing of whether it's antivirals or monoclonal antibodies. It really is about finding the patient. When will you find the patient during their illness? And does that correspond with the function of the drug that you're trying to give them? And so to me, one of the important things, and again, this comes back to connectivity here in clinical care as well, to diagnostics. It's easier if you have a product that's for prevention and you have a fixed population, for example, nursing home residents, as we've seen with monoclonal antibodies for prevention potentially, right? Or post-exposure prophylaxis. It's much more difficult in the big wide world where there isn't a captive audience, there isn't a recognized target organization or a population, because you need to be able to link rapid identification of someone who is sick, which is, hard when the drug is also potentially causing the bug is also causing asymptomatic infections. And then you need to be able to link it to confirmation of diagnostics, because you won't want to be able to give a drug after the confirmation of a diagnostics. And what we've seen here, if you'd asked me before this pandemic, I would have told you about just the immense, which already still exists, the lack of diagnostic capacity globally. There was a study, I think, in early 2010s about how 60% of malaria cases are still treated clinically. I mean, this is one of the most common infectious diseases in the world, and you come in there, you have the symptoms, you get a drug, you go home. And now if that ends up being an emerging pathogen, you've gone home and potentially continue to spread that. So I think that important connection to diagnostics is important, along with early identification of patients. The other bit is, the health systems. Again, I think the route of administration is going to be immensely important. And the amount of training and what level of healthcare worker needs to be present to provide that, here, if of course, but also internationally, can this be something that could be deployed in places that may not have the same kind of cold chain, the same kind of sort of capacity? Does it need the whole infrastructure of healthcare? Or could this be done in a more primary level, a community health clinic or something along those lines? Is going to really make a difference in terms of how high the uptake is. So I think those are the two things that I see are both opportunities, but of course also challenges.
Fantastic. And so I'm going to come back to this notion of stockpiles in just a second, but Dr. Bedelia kind of talked about this, the life cycle a bit, where we think about surveillance and we think about diagnostics, we think about vaccines, and this AHEAD 100 is kind of fitting in between, if you will, diagnostics and vaccines as we think about filling a gap when pandemics tend to happen. And so, Andy, the center is looking at this whole life cycle and trying to make sure that the fit of antibodies connects the dots, if you will. Is that, tell us about that approach a little bit and why you think that makes sense here as we talk about preventing pandemics.
Oh, absolutely. What we need is a system of good early warning, rapid characterization, and then based on those sequences, fast countermeasures. And so antivirals, monoclonal antibodies will fill that gap of, let's say, the first 100 days. And now we have a goal of vaccines in 100 days. So nobody has really, in a comprehensive way, been working on that area of fast treatment and prophylaxis. And that's the gap that this will fill. And by creating a consortium, it allows public funding and philanthropic funding and investment from the private sector to be combined in a very, very powerful way. And then we can exercise this system. That's very important. We can do real world exercises as epidemics are occurring around the world to prove it out, to overcome the regulatory hurdles in advance. And the goal with all aspects of this system is to reduce the times.
So that exercising is such a key part of what you've all have laid out for the center here, what we call the demonstration project, and basically proving out the systems that are yet to be proven with prevention. So Dr. Crowe, tell us a little bit about this demonstration project capability. What type of pathogen are we looking for? How are we going to prove the system out, as Andy says?
Yeah, I think this is very important to do simulations that are real, maybe is one way to think about it. So I think it's widely recognized in the current environment that the DARPA model, one of the US agencies that funds innovation, has been very influential about how to think about doing things that you can't do right now. And so they sort of say, well, our fundies came up with GPS and the internet, and now we're coming up with fast biologics. That was their goal. And we were tasked within an existing DARPA program called P3, Pandemic Prevention Program, to go as fast as possible. They wanted us to go from blood sample to having the drug ready to go in 60 days. And they actually put us through a sort of a simulated scenario, which we've been calling sprints. So you get a stopwatch and you click and you say, go, here's your blood sample, and then see if you can get it done in 60 days. And the first time we did that, we had never operated with a stopwatch. I mean, obviously, you always want to be efficient and fast, but having a stopwatch, in fact, our group was tweeting out live daily what was happening, and it is nerve-wracking because people were watching us, and that sort of upped the ante, including our sponsors at DARPA. And so I think we learned that you have to sometimes make decisions that are good enough in the moment to answer the question and move forward and get out of the high reproducibility, high precision, accuracy mindset of conventional science. It's a different way of thinking about how to respond. And we learned that ourselves. So we did kind of a fake round of antibody discovery with Zika. And during COVID, we were getting ready to do our second one. We were all geared up to do a bird flu simulation. And DARPA just turned to us and said, no more fake stuff. This is real. You need to go. And on a dime, we turned and did COVID. And I think if we had not done the simulated sprint the year before, we never could have responded. Whereas in COVID, we were able to do the antibody discovery in about 25 days and turn over antibody sequences to a partner. And now we're successful with these antibodies in phase 3 trials. So I think this idea of practicing, keeping a warm, ready technical capability, and not just doing it and stopping and resting, but constantly keeping the system sort of flexing. It's sort of like exercising. You know, you can get to a certain level of fitness, but you can lose that easily in about a month. But you know, it takes you months to get back. So you want to keep that fitness of the system high. And what we realized in the Head 100 idea is that many of these infections that could go worldwide are already occurring in the world. So, I mean, yellow fever right now is occurring in South America and in Sub-Saharan Africa. So if you actually had an intervention you wanted to simulate, it wouldn't be fake. You could actually go into an epidemic area and simulate pandemic prevention, but you'd actually be treating real patients in a real place. So we've been thinking about that, how to do a simulation of epidemic interruption that's real. You know, so to take the products or not just discover them, but move them out. And this gets back to what Nahid was saying. We know how to discover antibodies now. It's actually almost a turnkey solution. Blood goes in, antibodies come out. And yet, how do we get them in arms? Who do we give them to? And how do we even get people to accept them? This is a very complicated area that is the weaker part of the system now. I think in a Head 100, we want to, that's why we need all the partners involved, not just discovery and manufacturing, but we need the last mile, the last person, basically.
And so for the center, this notion of a demonstration project, the exercise, the going beginning to end is kind of a critical component of how the center is structured. But another key construct within the center is this notion, as Dr. Crowe says, of warm ready, of creating a stockpile of the top 100 pathogens in the world, right? A stockpile that can be deployed, i.e. that allows us to get on the other side of the outbreak, that allows us to be preventive versus reactive. And so that notion of a stockpile has been, I guess, looked at before in a variety of areas. Andy, tell us a little bit about kind of the role that the Department of Health plays in this, and SNS historically in stockpiling, and how this might be similar or different as we think about a global readiness capability.
Well, exactly, Stu. I see this as a pandemic insurance policy by having that preparedness in advance. After 9/11, 20 years ago, and the Amerithrax attacks, we invested very heavily in the Strategic National Stockpile. For example, we stockpile enough smallpox vaccine for every American. And essentially, we've taken that specific threat off the table. By doing so, we've signaled to our adversaries that smallpox as a biological weapon won't be effective if used against the United States. So we need to think in those same terms. Unfortunately, funding for biodefense writ large has been episodic. We go through periods of panic and big investment, and then neglect, and the funding atrophies. So we need to level that out, have a sustained investment in these capabilities in this system. And we really can take the whole range of biological threats off the table, whether it's deliberate biological weapons, accidental releases, or naturally occurring outbreaks.
The notion of prevention, it's amazing. And Dr. Bedelia, you've worked around the globe, and think about your special pathogens unit, stockpiling in global locations, and then administering and deploying those solutions as outbreaks occur. Harder from a process perspective or a science perspective, how would you encourage us to think wisely about deploying stockpiles in the heat of the moment?
Yeah, Stu, I'm going to answer the stockpile question last, because I think the challenges that I'll speak about sort of will address that. I think that I want to, those first 100 days that we're talking about, the benefit of antivirals or monoclonal antibodies that are broader, that are targeted to pathogens that we're interested in. The benefit is not just to the individual. The reason we want to get this right, the spectrum of diagnostics, surveillance diagnostics, getting person into care, is great for the individual's recovery potentially. But by getting people healthier, faster, keeping them out of the hospital. You are keeping hospitals from getting overwhelmed and potentially having secondary health impacts, which is what we've seen across the globe and here again and again during our surges. But you're also having an impact early in that outbreak or those 100 days in transmission. The quicker you can reduce the period of time that a person is contagious through potential medical countermeasures, you are reducing the opportunity for that person to continue spreading. So those 100 days are critical from multiple places if you can get it right. The difficulty that, one thing that monoclonal antibodies, at least from my side, from the healthcare worker side, the thing that I've noticed this during this pandemic is evidence generation for efficacy. You can have ready, monoclonal antibodies to go. But what I've seen is that it took, because it took that period of time to get that clinical data about where's a right fit, what patient, what period of time for illness. You saw hesitancy among healthcare workers in many cases in deploying monoclonal antibodies. And that really highlights the importance of a warm base for research. Often when we talk about stockpiles or readiness, we talk about the technological aspects of it. But the thing that we've seen that really is needed to go hand in hand with that is a research infrastructure warm base. Places like UK that did a good job of this with the recovery trial of being able to deploy already off-label products versus new products. They really were able to give answers to clinicians that they could deploy. And we in the US here, public health exists in the public sector and healthcare exists in the private sector. And when you need, this combined public health action, we actually need to think about what a research infrastructure and warm base here, as well as globally, will be. And that gets us to that point of once you have the stockpiles, are you going to be able to deploy it? And here's where the inequity part comes in. I think that we definitely could deploy it. I think that would require that extra layer of, global cooperation. And as we've seen from this pandemic, in some points we've been excellent about it, right? We've had trials, vaccine trials across the globe, and others we haven't. In the end, even the COVAX, which was shifting to example of vaccines, you had this COVAX utility to try to create greater equity and potential sharing of a monoclonal, of the vaccines. When push came to shove, you really saw a competition for products. And really, if we're trying to stockpile, the importance of this is going to be the ability to manufacture enough to get it to places where they need to be so you don't create that competition. And hence, it's the highest bidder who always ends up getting the products.
Awesome. And I think when we, as we saw COVID-19 unfold and we saw limitations on medical countermeasures, we saw limited supply, and we saw countries begin to lock down and not share, if you will. And we learned some lessons for that we're trying to all incorporate into the center to make sure that those countermeasures are available where needed, right? As stopping the outbreak is what's important. And if we can do that, then the rest of our interests can be well served from that point forward. Difficult challenge, as that's not a science issue. That's an issue of people. Andy, you, as we at the center, as we looked at the constructs for working something like this through the public sector side, you coined a new term. Tell us about that a little bit and kind of the notion of how you think about operationalizing an effort like a Head 100 as it intersects with the government.
Well, I've been calling for 10 plus 10 over 10. So an investment of $200 billion over the next decade, $10 billion per year for the Health and Human Services and its components, and $10 billion a year for the Department of Defense to focus on this biodeterrence and pandemic prevention imperative, a national security imperative. And I think with that kind of sustained investment, we can have the system, whether it's a weather map for tracking infectious disease, for early warning of infectious disease, and then the tools for rapid response, including rapid manufacturing, distributed worldwide manufacturing. We need to invest in innovation in cell-free technologies. So we can have manufacturing of nucleic acid products at the source where it's needed, and not have these inequities that Naheed points out that we're still, that are reaching tragic proportions as parts of the world still have not receive the first influx of vaccines. But that, with investment in a sustained way, we can avoid that next time. And we have to learn from our experiences and get better.
And so there are mechanisms today in place, like if we look at the antimicrobials, we think what was done with Carbex, and there are models in place where BARDA works effectively in a cross-sector fashion to make these things happen. Is BARDA the right instrument or the right area within our government to pull together those interests you just mentioned?
Well, I think Carbex is a really great example where it's not just projects, but it's an overall program. And that's why I came up with the term MABX, the monoclonal antibody accelerator, because I think we need a coordinated program like a Head 100, where we can, not just work on the one-offs, but have this comprehensive system and move the whole field of monoclonal antibodies forward as one of the anchors for this pandemic prevention system.
And so as Andy talks about the 10+10, and he talks about, all three of you, your lives intersect because the pathogen doesn't discriminate, if you will, in that sense, but public health and defense. And do those worlds really need to be collaborative and cooperating as we go forward? Or are they okay to operate more in a siloed fashion? Any of you, thoughts on that?
Let me just say, because I've lived at this nexus, they are completely overlapping, and we need to embrace the overlap and not get turfy about it. This artificial divide, whether we're investing too much in biodefense and not enough in emerging infectious disease, they are the same thing. And the example I'll give you is virus X or disease X. That could be a North Korean bioweapons program engineering a novel pathogen, or it could be the next, SARS-CoV-2 coming from nature. But what we need to be able to prevent and respond to that, it's the same thing.
And it's interesting, our current times. We're, and we've recently seen the report coming out of our own government that it's still inconclusive on whether SARS-CoV-2 naturally occurred or whether it came out of a lab somewhere. That's, regardless, though, the solutions are still the same that are required to address this. Is that right?
I think absolutely. We're talking about human beings. Every one of the target individuals is a human being, and the medical countermeasures we're going to use are the same. So it really comes down to these policy issues in my mind are who's going to own a stockpile, who has the ability to ship it or control it, but the actual technology is going to be the same. And so therefore the interests of the security community and the public health community are identical in terms of what types of countermeasures they need as a tool. How you use those tools is more of a policy, but the medical interests are identical in my opinion.
And I would echo on the public health side as well. I think the bioterrorism 101, right, is a pathogen that looks like other pathogens, doesn't make you, as sick, potentially overwhelms the healthcare systems, and all the things that you need for a naturally occurring pandemic that we just talked about: early surveillance, diagnostics, healthcare capacity, research, infrastructure to pick up the signal from the noise. It's all the same.
One thing I would say, we're at a moment when you think about what does it take to get this job done. Really, in all of the areas, we have all the tools to do the job, to be ready for epidemic. But we have not had the political will as a people, and I'm not blaming legislators. Legislators do what we tell them to do by our voting and polling and so on. As a people, we have not been committed to be proactive. We respond. Andy said, we have a panic, and then we just want it to go away, and we never want to think about it again. And that's just such a short-term mentality as a people. And and the question is, after COVID, are we going to change our mind, or do we just want COVID to go away and never want to think about infectious diseases again? And I think it's a turning point in human history, in political history, and social history. Do we have the consensus and will a people that we want to invest in this so that we're more ready next time. And I'm not clear that we've turned the corner on that decision of will. So we have the technology, but have we made the decision to be ready? And I think we need to speak to ourselves and our legislators and our families and friends. Like, we want to commit as a people to do this, and we're willing to pay the price, which is not a particularly high price in terms of finances.
We see some really interesting signs, I think, when within OSTP, within the White House, the formation of a pandemic prevention capability led by Eric Lander. I mean, that's new. That's being done at scale. That's to address this problem. That's incredibly encouraging. At the same time, as Dr. Crowe says, I mean, we don't have to look back far to the formation of CEPI. And we see CEPI having the global vaccine capability, yet we as a people in this country anyway, in the US, chose not to make that investment, right? And the Norwegians did, and CEPI has been incredibly, I think has been successful in driving vaccine attention. And now we've got that same opportunity with antibodies. Are we gonna respond differently at this point in history? One of the things that's encouraging to me as we all work through this center, this Global Pandemic Prevention and Biodefense Center, is that for the last 8 months, collectively, you, the other advisors, the team have put a strategy together for a Head 100 and to take it to market. The funding for those first 8 months was co-equally provided by philanthropists. Sachiko Kuno, the first one in, the Gates Foundation, Bill and Melinda Gates Foundation with their funding. We had a variety of industry players step up and provide funding. We had state and local government through the state of Maryland and Montgomery County step up and provide funding. So you had this public-private industry, academia, government coming together for this purpose. So Jim, I would say to your question, there are some good signs. We need to now follow through and make it a reality. We've just got a couple of minutes left. I'd like to turn it to each of you to for a last word as you look at this global pandemic prevention capability, this desire and this drive to go get in front of pandemics with medical countermeasures, in this case, the launch project with these human monoclonal antibodies. How would you encourage us all as a broad community, cross-sector community, to make sure that we don't leave the door open, that we actually drive to closure and get this center firmly in place, funded, operational, and get these exercises underway so that the next time we have an outbreak like the one we're living through right now, that we're able to put an end to it immediately. So a last comment from each of you. Dr. Crowe, we'll start with you.
Well, I'm excited that we're launching the effort, and I'm sure we can make very potent and broad antibodies that will deal with these infectious agents. So the time is now to do the technology, but can we come up with a strategy where this integrates into the world system of using them? And I think we're committed to coordinating with those who are in surveillance and diagnostics, in antivirals, and in vaccines to sort of prioritize targets so we get a mass effect where as each sector makes progress, the others come along with it so that we have all the tools for each of the agents. And so that coordination, I think, is a big opportunity right now. And we're asking partners to think about that, partnering with us and coordinating with us to be in sync with the prioritization schemes that we're using.
Dr. Bedelia?
Yeah, I'm going to echo a little bit of what Jim said, and I'll just add, F. Scott Fitzgerald said, I'm a cynical idealist. I'm definitely a cynical idealist when it comes to this, because I think we have made incremental improvements with every challenge. After the West African Ebola epidemic, you had these global setups, the CEPI, the other, global security agenda to try to bring some level of standardization for surveillance and public health preparedness and things like that. But yeah, we tend to cycle between panic and neglect, as Andy said. And the thing that worries me is that this is critical. We need to do this for anything else to happen, right? It is necessary, but it is not sufficient, because that coordination piece, the technology needs to go with coordination, as Jim said, but also that there are being clear-sighted about the fact that there are new challenges in this environment: disinformation, misinformation, state-sponsored disinformation. We talk about the crossover between state and public health, the state policies in public health. These are all new challenges that didn't exist maybe 20 years ago as bad as they are now that we'll have to continue dealing with.
Super. And Andy?
Well, I would just conclude by saying we have a once in a lifetime opportunity to get this right. And you know, I was in my office 20 years ago in the Pentagon when we were surprised by the 9/11 attacks. We can't be surprised. I mean, we saw this pandemic coming, and now that we're understanding the costs, let's seize the opportunity and make those investments so it never happens again.
Awesome. Well said, all. Thank you, all three of you, for your sponsorship and support of the Global Pandemic and Prevention and Biodefense Center. Wish you all the speed and success in the world in that. Thank you for your time to tell that story today to this audience. And with that, we'll call this closed today. Thank you.